My name is David Fairlamb. I'm part of the clinical team at SolasCure, Director of Regulatory Affairs, that is developing Aurase Wound Gel for the treatment of debridement for venous leg ulcers. So I think the understanding is that debridement is very key to good wound healing. And without good debridement, healing can be delayed. Unfortunately, there is no consistent standard of care across the world. On one hand, in certain markets, surgical debridement is very good. But actually, outside of the US, for example, most patients simply are given moist wound healing dressings, which utilize autolytic debridement, which is very slow. And so they get slow debridement and slow healing. So I think the lack of standard of care is certainly a major unmet need in this area. And I think also recognizing that these patients are largely in the community. So we need products which can be used very easily, which facilitate the nurse's application of the product. Products that can be used not only in hospitals, but equally they can be used outside of hospital, in clinics or in the patient's own home. And a product that fits around the standard of care, dressing changes that these patients are going to get, and obviously something that doesn't affect the viable wound bed. So I think all of these factors come into play with venous leg ulcers, and it means that patients simply don't have good, consistent standard of care. So our enzyme is Tarumase. It is a enzymatic protein that was first identified as a key component, an enzyme in maggot saliva. Having identified the enzyme, we cloned it, and we now produce that enzyme recombinantly. It's a fibrinolytic selective enzyme, but it does have action on collagen, and it does have action on elastin as well. I think Tarumase itself is actually an excellent enzyme from which to base a development for a chronic wound debriding product, because it has been developed itself through evolution. We're not trying to recreate something. We're trying to use what nature has already produced. This enzyme isn't painful, and it's not immunogenic. And those are two key characteristics that enzymes, particularly ones that are used for debridement, have historically had issues with. And so it's a factor that we want to address. Now, we've taken this enzyme and we put this into a hydrogel. Hydrogels, as you know, are commonly used as a means of producing autolytic debridement. And so what you get with Aurase Wound Gel is a synergistic effect between the activity of the enzyme, so the faster debridement, but you get the moisture benefit from the hydrogel, and that is pro-healing as well. So I think together, we hope that this product will have an excellent profile for use in wound debridement. So this was the first clinical trial of the product. It was therefore designed as firstly and primarily a safety study. We sought to do this in an open design, where we could escalate the concentration in serial cohorts, increasing the concentration of Tarumase to assess its safety profile in this venous leg ulcer patient population. Obviously, this was a patient population. These patients did have sloughy wounds, and therefore, not only were we able to evaluate its safety as we escalated the dose, but in addition to that, we were able to assess the efficacy of the product as we increased the concentration of the product. So the whole point was to demonstrate safety and to provide proof of concept in a relevant clinical model that the patient that the product has the capacity to debride these types of wounds. So as I said, it was a safety trial first and foremost. So we looked at pharmacokinetics, so the systemic absorption of this molecule from the wound environment. It's applied topically into the systemic circulation. We also, because it's a fibrinolytic enzyme, looked at its ability to influence or have an effect on the systemic clotting process. And we looked at a number of other systemic safety factors. We were pleased to see nothing we could detect systemically, and there were no adverse effects in the systemic circulation. We looked at it from a local tolerability issue. So we looked at erythema, edema. We looked at wound itch and pain. We looked at induration. We looked at the level of bleeding, and we looked at the rates of infection. And again, had no particular concerns in relation to any of the local tolerability aspects. Then as exploratory measures, as I say, although it was a safety trial in this venous leg ulcer patient population, we were able to look at the ability of the product to debride, its rate of debridement, and we were also able to look at healing. What we saw in terms of debridement was that as you increased the dose or the concentration of Tarumase in the gel, you got a corresponding increase in the rate of debridement. So we saw more people achieving greater levels of debridement, but we also saw that that was being achieved at a quicker rate at the highest dose in the clinical study. And in terms of healing, we also saw in line with the debridement action that as you increased the concentration of Tarumase, you also increased the linear rate of healing, and you saw an increase in the percentage of granulation tissue. Well, I think there is an opportunity to look to even higher doses to see whether the even higher concentrations have an ability to further increase the rate of debridement. And in addition, whether these doses have an ability to influence not only the rate of granulation tissue, but whether that ultimately relates to a faster time to complete healing. And so these are aspects of the study we need to look at. Clearly, we need to move on from an open study, and our next one will be a randomized controlled trial. We need to control for various factors, like the age of the patient, the size of the wound. So we'll be looking to run our next phase 2 clinical study, which will seek to employ a number of these factors to look at greater emphasis on efficacy in terms of wound debridement and healing. We are keen to discuss these clinical trial results with anyone that wishes to get in touch. We would like to ensure that there is an opportunity for patients to get access to these new products. And so please feel free, if required, to get in touch.